Hello and welcome back to our Researcher Behind the Image series, where I, Jess, your Antibody Society representative, interview the researchers that submitted into our 2025 calendar, which is downloadable from our website if you haven't managed to nab yourself a copy yet. I'm super excited for this month's entry, as Danielle was part of last year's inaugural calendar, and I'm looking forward to hearing what herself and Fortis Life Sciences have been up to since we last spoke to her. Thank you so much for joining us again, Danielle.
Mhm.
And what is your position at Fortis Life Sciences?
Right, so I am the spatial biology manager here at Fortis Life Sciences, and I'm going on roughly 6 years here.
Brilliant.
Mhm.
Spatial is becoming such a hot topic. You see it around a lot of places. So I'm really interested in learning, particularly what you guys are up to, but learning more about the technique. What type of platforms does Fortis mainly focus on?
Right, so we have a couple of different instruments. We have the PhenoImager HT from Akoya, which is a typically used traditional TSA methods, like an
Okay.
opal dye. And then we also recently acquired a Comet 1.0. So we've been working with Comet for about 2 years. And so about 6 months ago, we finally got a 1.0. And so we're pretty
Oh,
excited about it.
congratulations.
Yeah,
I'm sure it's always fun to play with some new
it
toys.
is.
And well, this image is longer than 6 months old, so I'm guessing that was using the former apparatus.
Right. So this is actually generated off the PhenoImager. So prior
Yep.
to um, obtaining the Comet, we've been working with the PhenoImager since I arrived here in 2019, and this is one of the beautiful images off of it.
All right. Would you be able to take us through exactly what we're looking at then?
Yes, yes.
So here
Okay,
we
great.
have our fabulous June 2025 image. Thank you.
Yes. And so I titled this an evaluation of proliferative activity. I also am looking at 𝗗cadherin and beta-catenin expression. Two of those
Mhm.
markers I've actually had quite a bit of history with. I've worked with them since I arrived here. And so
Oh.
this is a bit of an exciting little side project.
It's always
To
nice when it actually pays off. You said 6 years, right?
Yes, yes,
Wow, long arch, long story.
absolutely. So I wanted to just sort of recap. I wanted to share a few of the newer things that our site in particular
Yeah.
has sort of branched off into. And we've been an antibody manufacturer for well over 30 years at this point. And we've kind of always done custom antibody services, but we've really started to grow in that area. And
Yeah.
so I wanted to highlight that that is something that we are really focusing on moving forward. Of
Well,
course, we
that's
offer
fairly
several other.
relevant.
Oh, yes, absolutely. We also offer, of course, polys and monos, but the custom service is something that's really blowing up. And then just a quick overview of our approach to validation, because that's another extremely hot topic along with spatial, at least in the antibody space.
Yes.
Um, there's so many antibodies on the market that are not as great as they could be. And I think part of that is just the way that we can improve on validation will limit some of that. Um,
Yeah. Yeah, definitely. So yeah, so you guys have quite a rigorous QC from looking at this slide.
yes, we really wanted to sort of build our validation process around the international working groups pillars. And so we came up with our own sort of system, and you can, excuse me, you can see that here. Um, it is quite stringent. And I actually, to showcase our process, I recently did a poster over it at AACR,
Okay.
which went over very well. I utilized a different marker there. You can see it was PAX8,
Yep.
which is another fairly hot marker in the space right
Yeah.
now. And just rolling back a little bit on the actual process to get to the image. In
Yeah.
our lab, we go from actual fixation of tissue or cells all the way through embedding, sectioning. You can see that there's a tissue microarray, but we also construct cell microarrays, which is what was
Yes.
used to generate the image.
Yeah, well,
And
this
that,
is a huge part of spatial. Getting the quality of the tissue to start with is a huge hurdle. I've heard. I haven't personally got experience with spatial yet. I would love to. But I hear that that's a huge hurdle in actually achieving a lot of the spatial data that you see. The amazing data coming out there is highly limited by the quality of your sectioning and things going in. Do you have any advice for people who are starting out on those sort of histology steps beforehand that you've noticed as part of your experience?
right, yes. So the most important step is proper fixation of the
Yeah.
tissue. I know that we primarily work with FFPE,
Okay.
but fresh frozen is something that is not something I can really speak on too much, but improper fixation, whether that be the actual fixative agent or time, or even sometimes the age of the sample, it all
Yes.
plays a role in specifically the autofluorescence issues that I come across.
Yes,
Um,
that was what I was going to ask when we spoke later. That is a big problem, and do you notice it more in some tissues? But by the sounds, if you fix it correctly, it gets around a lot of that.
Yes, it occasionally, I think liver gives me quite a few problems, but really if you fix it appropriately and you use tissues or cells that are not extremely old,
Yes.
you will generally have that lessened. It also plays a role in just the ability of the slide and the tissue or cell, like the ability to stay on the slide. Sometimes,
Okay.
if it's improperly fixed and you try to stain, it will just come right off.
Oh, okay. That I didn't predict that one happening.
Yes,
Just come straight off. That sounds like a bad day in the lab.
it's a very bad day. But I wanted to get quickly into
That's
the,
beautiful.
yes. So I wanted to get into the PhenoImager. This is
Yeah.
obviously not the image generated, but this is something that was generated a few years ago on the instrument. And this is the instrument used to generate the image for the calendar. We've had this instrument for about 10 years, and it's really quite wonderful. As of recently, this week, or really a little bit late last week, we have onboarded the Parhelia spatial station, which is going to really enhance our efficiency using this instrument because it will be a new auto stainer so that we can couple
Oh.
the auto staining process with the imaging process.
That would save so much time.
Yes.
That is brilliant, the toys that exist out there.
Oh, there's
I'm
so
very
many.
jealous of you getting to play with it. So if you could, how many times do you restain or like go through this staining cycle? Do you find that there's a limit to how much a tissue will take, or does it depend again on the actual tissue starting out?
Right. So traditionally, this particular method, you can use up to 6 markers
Yep.
plus DAPI, of course. I would say around the fifth or sixth antigen retrieval because you're having to heat it so often that the tissue really does start to feel that. Especially if you're using extremely tiny microarrays, you may even see
Yeah.
that portions of them or the more delicate tissues start to fall off, which is, it makes part of that difficult.
Yeah, yeah, no, that makes sense. That's still like 40 to 50 or so different things you can look at once. That's
Hmm.
pretty impressive.
Yes. And although this instrument was not used, this is the other instrument I spoke about earlier on where we
Yeah,
just got the 1.0 version. You can see that in the upper left corner. And
yeah.
another exciting thing happening right now is that this instrument, along with, so Lunaphore partnered with ACD Biosciences, and they have offered sort of an RNA-scope technology. So it really has opened up the door into multiomics on this instrument.
Yeah.
We're very, very excited about it.
I can foresee next year's image
from
this slide. So
yes.
Tune in next year, hopefully, for to hear more about how this is going. But that opens up a lot of doors. How are you at Fortis looking at optimizing this? Like, what avenues are you going to be looking at, if you can say?
Yes. So we are going to do, of course, our own in-house sort of research-based projects, but really we want to target any customers that are into multiomics. I think that there are a lot of people that are interested in the combination of protein and RNA, but I don't feel as it has exploded to quite the degree that just spatial has at this moment.
Yeah. No, I think for a lot of people, they're curious but find it quite intimidating. So
yes.
I'm sure they'll be interested in hearing how you've incorporated the two together and how successful it is as a platform, because I'm sure it could be applied to many people's research, but knowing how successful it could potentially actually be and how viable it is as a platform is another thing aside from just being exciting to do.
Right, exactly. Yeah. So getting into the actual image, we used MCF-7 cells. On site here at Fortis, we keep several lines going.
Yeah.
And then we introduce occasional lines when we're trying to validate new antibodies, but MCF-7s are very easy to keep, or I should say relatively easy to maintain. And of course, they're used across a wide variety of research applications. So it sort of made this a little bit more applicable to a variety of different areas. I really personally wanted to observe the 𝗗cadherin-beta-catenin co-localization. I've done studies in previous years over this in tissue, specifically breast cancer tissue. So it was really interesting to me to be able to visualize this in cells.
Okay, and was this cell line picked because it has a lot of expression of your 𝗗cadherin, or is that ubiquitously expressed through all of your cell lines? Was it easy to see?
So this cell line in particular was chosen, of course, for the aforementioned reasons, but it also it can have sort of a gradient of expression when it comes to those two targets, which is why I introduced the 𝗗₃⁶7, because it was expected to be pretty proliferative and ubiquitous across the cell line.
Yeah, no, well, that's showing up strongly.
Yes. Yes. I would like to dive in more. I use so many different tissues and cells in the lab. We're not really a focused area. So, but breast cancer is something that's a personal interest of mine. So I often go back and into various tissues and cell types. So this is why I would like to evaluate a few more targets like ACE2 and ER and PR, so forth. And then eventually, we would like to evaluate cell lines a little bit more in depth on the Comet. We've done some sporadic studies on the Comet with cells, but we haven't really had much of a, I don't want to say much of a reason, but a lot of our customers that utilize the Comet are using it for tissue-based assays.
Yeah, so not many have used it for a cell-based assay yet.
No, not yet. But maybe some images generated off of it will get it rolling.
Yeah, no, that's exactly it. So what would be the advantage to using the Comet? I mean, I know it's more in depth, but for people who aren't as familiar with the machines, what would be some improvements you would see by using it on the Comet?
So I don't know that it would necessarily be more in depth on the comet.
Mhm.
I sort of worded it that way to give it a more like several more layers. I'd like
Yeah,
to dive in on the comet with more targets because
yeah,
unfortunately the PhenoImager, you have a bit of a hard stop at 6. But if
okay.
I take this cell line on the comet, I can then evaluate, 20, 30, sometimes
Yeah.
even 40.
Wow. Yeah. Jealous of being able to have that many antibodies for sure. No,
Yeah.
that's going to be very hard to sort of visualize. Have you, with your experience on the comment, how have you found the analysis side of this? Because I know that there's something a lot of people moving into it. You do need to learn new techniques and things like that to be able to visually represent so many colors at once.
Oh, yes. Yes. So with any challenge on the multiplex side of things, everyone gets very excited about numerous markers, but then of course when you overlay them on top of each other, they're just give this horribly
Just looks like
white
a
blob.
kaleidoscope.
Yes.
Yeah.
So it's oftentimes people will showcase the image in groupings, so it will make it
Yes.
easier to visualize. And so I think you really start there when you begin to analyze. You break out your nuclei first and foremost, and
Yeah.
then you take your image analysis in groupings of maybe 3 to 4.
Yep.
I do not want to speak too much on the analysis side. We actually partner with um, 𝗗𝗶𝗶𝗄𝗃𝗃𝗃.
Okay.
They're very recently rebranded from PathoMix.
Okay.
Um, but yeah, I know that the Horizon software that Lunaphore offers, and I'm hoping to get my own um, trial run of the software. It actually is super user friendly, and just getting to see some demos at AACR, it does segmentation um, at like the click of a button. So it
Brilliant.
makes basic analysis for users quite easy.
Yeah. And so, as someone starting in this field, do you have some advice for people trying to work with these sort of platforms? Like, what did you wish you'd known when you first started setting up these larger multi-omics sort of platforms?
Yeah, so I've had the pleasure of working with so many different platforms, and they each present their unique challenges and then of course
Yeah.
their, pros and cons sort of, so to speak. Um, Whatever instrument you end up having access to or maybe are looking into, just really I would suggest to focus on what is most important in the lab. Um,
Yeah.
and then the technical expertise of who you're working with or who you're working for, because there are some instruments that require conjugation. There are some instruments that do not. There are instruments that stain and image the sample. And then, of
Yeah.
course, there are some that are just imagers. So it's really, really dependent on what you have already. And then what, of course, your staff and your colleagues are technically capable of. I mean, if you have a great group of conjugation scientists, absolutely go for something like 𝗁𝗵𝗃𝗃 or
Yeah.
something like that. But
Yeah.
the
And with
Comet's been
your,
great.
oh, good.
Yeah.
It's nice to get people's opinions on how things are
hmm
working. With your customizable antibody service, do they take into account like the conjugations for these sort of platforms as well then?
Yes. So our custom antibody services, I'm not entirely sure on the, percentage of customers that come needing an antibody for a specific platform.
Yeah.
Typically, they're just looking for a target that may not be very prevalent out in the wild, so to
Yeah,
speak.
that's exactly
Um,
it. But say someone watches this and they had something that's been epic failure, like what should they be asking for when they come to someone like yourselves?
Right. So honestly, I would suggest if you're a customer or somebody watching this video and you're struggling with a particular instrument or a particular target, just reach out. There's almost 100% chance I will be the one on the receiving end answering the questions, and sometimes I can just troubleshoot a very small area of maybe where things are going wrong, and if I can't, then we can bring in our scientists. We have several on staff that we answer emails and troubleshooting requests daily.
Communication is key, ultimately.
Absolutely. So I just really want to reiterate that again, if you have any questions or concerns. I know that spatial it's not new per se, but of course it's exploding in the field, and there's just so many choices and so many instruments out there that it can get overwhelming, and it's hard to choose what is best for your lab or what's best for just, you know, your institution, honestly. Because a lot of these things are quite they can be applicable across so many different assays, and
Yeah.
I would suggest to really just look into, again, you know, your technical support that you have, your staff, your what the questions you're trying to answer. All of those play a role in the right instrument for you.
No,
There's
definitely.
no one right answer.
Thank you so much, Danielle, for joining us again. And a huge thank you to all of you for your ongoing support as part of this series. I look forward to talking to you next month for our July edition of The Researcher Behind the Image. So see you soon. Thank you, Danielle, and everybody for joining us here for this June edition. I will see you soon at the next episode. A huge thank you to Danielle and Fortis Life Sciences for their ongoing support of this series, and to all of you for tuning in and listening to what our researchers are up to. I hope to see you again very soon in our July edition. Tune in soon.